Change in the DNA molecules can cause mutations. One base pair is exchanged for another in the DNA molecule. One or more base pair inserted in the DNA molecule. One or more base pairs are deleted in the DNA molecule. There is a rearrangement of sections in the DNA molecule. There is an exchange of DNA region with another DNA molecule. That's recombination. So some mutations are harmful, some beneficial, and some are neutral. So what makes it harmful? Harmful mutation may cause genetic disorders or cancer. A genetic disorder is a disease caused by a mutation in one or more or few genes. It is generally caused by mutations in genes that regulate the cell cycle. Because of the mutation, cells will damage, DNA are allowed to divide without limits. I've read an article about a breast cancer woman in the America, and she has a second and third opinion already because she wasn't satisfied at first, because the first thought her that it was just about the lumps in her breast is just a lumps clog that's a result of after pregnancy, but she wasn't satisfied in it, so she had a second and third opinion of an OB/GYN. She have visit some far away hospitals from her town. So what she did is, in her second, she wasn't still satisfied, because same goes by as the first one, that it was just a result of after pregnancy. So in the third one, it refers to have a genetic testing. I don't know if that is present here in the Philippines, but in America they have a genetic testing, because she have shared that in her history about her mother, that her mother had been a breast cancer. That's why they have recommend to have a genetic testing, and that's it. It was some sort of positive, or she has they have explained her that it was good that she had a genetic testing, because it was quite, there's a connection of her history. So I know we had already heard about this, that but she have written, or she have shared in her article, that there's a quote there that she says, "Knowledge is power. The more information you have, the better." Because she was always curious about her health, especially at her breast, because her mother died at a very young age because of breast cancer. So she wasn't satisfied with what the OB/GYN had given or had said or explained to her, so she had up to third opinion, and that's it. She had her unanswered question. So let's proceed to plasmids. Some plasmids encode for genes that enhance pathogenicity of a bacterium. E. coli plasmids encodes toxins produced and bacterial attachment to intestinal cells. Transposon are small pieces of DNA found in chromosomes and plasmids. They are able to direct synthesis of copies of themselves and become incorporated in the chromosomes, called jumping genes. Because of their ability to insert themselves into our chromosomes or change their locations. And lastly, transduction. Transfer of genes from a donor to a recipient by a bacteriophage. So this pictures is a very basic explanation of mutation. So the original copy and the mutant copy. So if it wasn't really 100% that he really replicate the original, so what makes it harmful if he replicates the abnormalities of what has the original. So that are some of the very basic explanation of the mutation, that original and mutant. So that's it. Thank you.